specimens. We have the Polygonaceae fructuosa, better known as the petite butterfly, and the Polygonaceae, better known as the violet tree. What happens? Both of these specimens come from the same family, the Polygonaceae, better known as milkworts, and moreover, they are believed to come from South Africa, both of them. So they have the same origin and the same family. Nevertheless, we have the Polygonaceae fructuosa, which sizes up 2 to 3 feet, and the Polygonaceae sizes up 60 to 65 feet. So what made this tree grow under such conditions? Now, so you get a little bit of the background. We have the Polygonaceae, which is the main family. They are better known as milkwood or snake roots, which they're known as milkwood or snake roots because ancient Greeks used to give them to the cows so they milked better. So, as you all know, cows can't climb trees, so they needed to feed from the shrubs, right? There are approximately 17 genera, on which there are about 900 to 1,000 species, on which half of them are shrubs. So what happened to the Polygonaceae? That's exactly the purpose of our experiment, in order to sequence and analyze what makes this stand over other shrubs in the family. Now, we said that they come from South Africa, believed to come from South Africa, although the most abundant proportion of the population is believed to come from South America and Asia. Now, it is believed that the Polygonaceae arrived in Puerto Rico on September 22, 1998, after Hurricane George. I quote Ben Blumano when I say, "This natural disturbance had an effect on the phenology of rare tree species, for example, Polygonaceae flowered conspicuously after this event." After our visit to Ayunque and talking a little bit with Professor Velasquez, he told us that the Polygonaceae rise in Ayunque in order to avoid soil erosion. The Ayunque needed it to grow, so it grew. It helped avoid soil erosion, although it grew excessively and fiercely. When it started growing so excessively, avoiding all natural competitors, such as termites, which help balance the ecosystem, federal forces had to take action in order to mitigate these trees for other species to grow. Now, how will we study a tree? We'll use bioinformatics to measure the difference between common genes in the Polygonaceae fructuosa and the Polygonaceae. We'll need 3 samples from the Polygonaceae fructuosa from Puerto Rico and from South Africa, because they're origins, and we'll need 3 other samples from the Polygonaceae from Puerto Rico and South Africa in order to sequence their genes. Now, how will we do this? A process named RNA-directed DNA methylation. It's a long name, but in terms of complex, it's not that difficult. Okay, RNA-directed methylation is basically acknowledging changes or adaptations on parent strands to daughter strands after replication in a process called de novo. When we study methylated DNA, we study silent genes in the structure. Now, when you find silent genes in the structure, you might discover diseases such as cancer. Moreover, when there is change in gene structure, it doesn't necessarily have to mean that the plant is ill. It may mean that it adapted and it evolved. We all know natural selection, right? Now, we'll study as well the polygenic inheritance. As we all saw in class, polygenetic, in which a species has more than one gene in its sequence and structure. And there are about 180 genes which affect height. So the thing is, when we see genes that affect height, we see plants that grow taller than others in the same family. Now, we'll as well study consumption and latent chemical energy of this tree compared to others, and the resistance to termites and other organisms which help balance the ecosystem. We'll make a cytogenetic map. A cytogenetic map is basically the study of a gene structure. We'll compare and contrast gene structures from the Polygonaceae fructuosa and Polygonaceae. Now, this is a kit we'll be using, Easy DNA Methylation Kit, provided by Golden Gate Infinite Assays. And I show this equipment because it is one of the best classified in order for this study, to study good DNA and PCR for future studies. So thank you very much for your attention. Any questions, suggestions will be appreciated.
What will be your future work?
This is a great question because, as we all know, you can't see trees growing so exponentially, but I prepare a slide especially for that. We have future experiments on the iguana. As we all know, the iguana is an invasive species as well. And the main point for this experiment is to study island conditions. How ideal are they for Puerto Rico? As we all know, this little guy has grown exponentially in Puerto Rico, and we may study its sequence in future studies, such as its pineal gland, which is this thing is between the eyes, called the third eye. What happens? In Puerto Rico, it may be assumed that it has not adapted because it doesn't have natural hair. But that's another study. But thanks for the question. Any other question will be appreciated. Well, thank you very much.